again, because what you see on the screen there, you can actually see, and the screen's big enough and you can see it, is you see just diffusion occurring. And in this case, they've made a container that has a hole in the center. And so the only way that compounds move through here is by diffusion. So in reality, what you've got going on is water moving this way, and then the solute, which is what's solubilized in the water, moving through the hole until basically you get this random arrangement of molecules just by movement, Brownian movement, just the fact that the molecules are moving around. And this line here, because we're going to be talking today about how we can do some work with this, it's always going to be the same, okay? So water is moving back and forth, but in terms of energy, which we're going to be talking about today, more water is going to move from the higher concentration of water to the lower concentration of water, okay? But we're not dealing with a membrane yet until we get to this equilibrium state. Now, with osmosis, we now have this theoretical membrane that only allows the solvent, in this case water, to move through. So, as we're going to talk about today, water potential, in this case the potential of pure water would be 0. And over on this side, and we're going to go over this, the water potential would be less than 0. So water's going to move across, and the big thing here is water's going to move. There's actually energy involved, and it actually raises the level of the solution above the level on the other side. So in this case, they're always going to be the same. And so in this case, you've got the fact that there's a difference in the potential energy of water driving this to move this column of water and solute up until it gets to the point where gravity keeps it from going any higher. And after we finish, if we get through the end of the water potential today, we're going to go back and talk about root pressure. So I think we finished up with the fact that we've got these sets of membranes, and if we look at the cell wall and what we have going on, in this case, here's the smaller vacuole, here's the chloroplast. So you're going to have water moving in all possible ways. But basically, we talk about moving water from outside the cell to inside the cell, okay? And in fact, the vacuole is the main driving force. So the water potential outside the cell versus basically the water potential of the vacuole. But we just talk about it as inside and outside the cell, outside of inside, okay? Now, this is a chemical potential. And what's chemical potential? It's the ability of something to move. So if we're talking about something moving, we're also talking about energy. We're talking about work, whatever work is. Work is actually doing something with energy. And in this case, we're talking about water. And we know that the chemical energy of pure water is 0, and it's usually seen as psi, though it can be different things. It can be W, it can be several different things. And the take-home message here is anything that is not pure water, any aqueous solution that has a molecule completely solubilized in it, and by solubilized I mean if we put sodium chloride in water, the sodium and chloride separate and they're completely surrounded by water. then that's going to lower the water potential. It's going to lower the concentration of water. So you could write down that, pure water is 0, and everything that we're dealing with is going to be, the water potential is going to be much less than 0, always. The only time we have a water potential of 0 is when we take a leaf, put it on a slide, and put distilled water on it. That's the only time when the outside of the cell would be 0 too. Okay? Now, water is going to move through the membrane passively. Now, what I mean by passively, it means that water movement is not energy dependent. So if you think back to those transporter proteins, and some of those transporter proteins required ATP, the active transport proteins. What you're going to see is there are actually proteins that speed movement of water across the membrane, but water moves without energy, without burning ATP, basically, and that's what that means. Now, what can move is, by active transport, is ions. And probably next time I'm going to send you out a few slides where we're going to talk about something called osmotic adjustment. And that's something that a plant can do when it's dealing with water stress, like it's becoming more and more common. Now, what does that mean? So, let's say we have a nitrate that's moved into the cell by active transport. So every time a nitrate moves in, what does that do to the water potential of that cell? Goes down, becomes more negative. So what plants can do, and we're going to go over this again in another lecture, is if there's the concentration of water is lower outside, then the plant can actually pump in ions, which will lower the water potential inside the cell. Okay? And so water can still go in. So in some plants, they're able to what's called, and we're going to go over this again, osmotically adjust. So if you take a plant that is, let's say, will grow okay, will survive, let's say, high salt levels, and I mean survivable salt levels in the water, then that plant can sometimes pull those salts inside. And so by pulling the salts inside in the vacuole, it lowers the water potential to the point of where the water concentration is higher in the salty water, and the plant still grows. Okay? I'll be sending you out some slides about that later, today or Wednesday. Now, let's go back here to our osmometer, and I think you can still get these. I should get one of these. And we're going to go over several different slides where we're looking at the osmometer, because it's a very easy way of introducing the concept of pressure and water movement and the physics of how water moves. And in this case, we've got this thing called a thistle tube, which I remember from high school, with a membrane on the bottom of it. Okay? And inside this glass container, we have a solution. Okay, a solution means it's water and something else. Outside, we have pure water. So what's going to happen when I take this thistle tube and I stick it in pure water, which way is the water going to go? Water's always going to move from higher concentration to lower, so we have water moving in, and we got some work being done here. So, in the last slide I'll show you is where there's actually an osmotically driven power station being built. I can't remember where in Europe it's being built. So, if you think about this, it's not a very fast process, but given enough change in water potential, you could use this as a lift to lift a car up. It'd probably take quite a while for the thing to come up, but just to give you an idea. And so then what we can do is then we can actually put a piston in this and we can drive it down, okay, to where we're at this equilibrium site. Now, so we can actually push the water, and that's going to take pressure to push the water down, right? And that's another component. Pressure potential, which we're going to talk about, that's what causes the cells to be blown up against the cell walls, the pressure. So given that, this is an example of something that you see all the time. Probably a lot of you have these in your home. What is that an example of? A very common process, especially around here.
Water filter.
What kind of water filter? Reverse osmosis. What's osmosis? It's movement of water from higher to lower concentration. So what must reverse osmosis be? Moving the other way. So on the right-hand panel there, you see if you start at the top and you put pressure, what's going to move? Only pure water is going to move out of that membrane. So if you put pressure enough on the membrane, you're going to take that solution and you're going to move water out of the salt water into the pure water by putting pressure. That's what reverse osmosis does. And it's been around since, I think, since I was in high school. So you're putting pressure and you're forcing now the water to move. Now let me keep this in mind, that at any given time, water's still moving both ways. It's the relative movement of water. So if we got this beautiful turgid cell, theoretically, for every water molecule that moves in, one moves out. Otherwise, it would explode. And this pressure now is called osmotic pressure, okay? Pi, and it's also called P too. And you'll see this both as a pi and P. Now, we have another term we call psi sub s, which is the osmotic pressure, okay? So here we actually have a pressure, an osmotic pressure, which is due, or solute potential, solute potential that's due to what's solubilized in the water. So in this third one, again, is we're just looking at the pressure, and the point here is that we can adjust the volume and we can put a pressure. So at this point, the pressure potential, when it's right here, is going to equal to the solute potential. Okay, so we can actually put a pressure on here until we get exactly back to that line, and they're going to be equivalent. Now, why are these important? Because this defines how water moves. So we have probably more people in this state measuring water potential than, well, probably California now with the drought there. Probably more people here in California measuring water potential and pressure potential than any other place, because it's so important to plant growth. We've gone through this massive drought. California's gone through this drought. And these terms are how we describe the physics of water movement. Okay? Isn't there any of you ever measured water potential? There's several ways of doing it. Measuring pressure potential, there's all sorts of instruments. And it gives you a snapshot of what's actually going on in the plant. Now here's an equation that you will need to understand. The water potential, and this is sometimes psi sub W, is the pressure plus the osmotic potential. Okay? Pressure can be positive or negative, and we're going to finish up, if not today, on Wednesday, talking about root pressure again. And solute potential is always negative. When would pressure potential be negative? We've talked about it several times now. When is pressure potential negative in a plant, and where is it negative? Usually.
In the leaf, like coming out of the...
Coming out of the leaf, in the xylem. Remember, water is sucked up the xylem. Part of the way it's pulled up, so it's got a negative pressure. So xylem is under, usually the pressure is negative. Think about it. Otherwise, there wouldn't be any suction on it, right? So remember, in that xylem, we've got, and it's not really negative, but remember, we've got a column of water that's sort of being pulled up. Solute potential is always negative. If you have pure water, it's 0. If you put one single molecule in that pure water, it now has a negative, slightly, very, very slightly negative water potential. And usually when we talk about these components, we talk about something that's surrounded by a membrane or as part of the system has membranes. Okay? Now, let me mention another term that we're just going to mention. It's important. It's actually really important in this part of Texas, called the matrix potential. And that only comes into play when we're talking about seeds and seed germination. And the matrix potential is due to the makeup of the dry seed. Okay? What's the water potential of a dry seed? Anybody want to guess? It's like minus 1,000. You know, it's made up of carbohydrates, starches, lipids, proteins, that all will suck up water at a different rate. Why is it important in this part of the world? Most of my work had until 10 years ago been on seeds. So upstairs we have a seed storage room on the 5th floor where we control the humidity and the temperature. Because if you have a high humidity area, if I take seeds out, put them outside in the summer in Texas, the water potential is so low that they actually pull water into the seeds from the atmosphere. Not enough to germinate, but actually enough to kill the seeds. And even inside where we have air conditioning, to keep seeds from degrading, we have to store them in this seed storage facility. So we control the humidity, we lower the humidity so that we lower the gradient of the water potential, and of course we control the temperature. When I was in Washington State, they had the state seed storage facility on campus. They just had the seeds laying out on the counters. They had them in just normal shelves because the humidity there is like 10%. So matrix potential is important, and it will kill seeds. You will not pull enough water out of the atmosphere to get a seed to germinate, but you will pull enough water out of the atmosphere to actually get the seed to start to do something and die. It'll actually start trying to reform membranes. So in humidity and high temperatures, matrix potential is something that's very, very important. Also, one last thing, and you may have seen this experiment, you can take seeds and you can make up some plaster of Paris, and you can just make a big wad of plaster of Paris and seeds, and what happens? Anybody ever done that? Or you heard where a seed falls into a crack in the cement? What happens? If you take seeds and put them in sort of a wet plaster of Paris, they will start pulling water in from the plaster of Paris, and the seed will start expanding, and the plaster of Paris will start, blowing apart, not blowing apart, but cracking apart. Okay? Think about the force involved in that. Uptake of water into a seed has nothing to do with whether the seed is alive or dead. It has to do with the matrix potential of the seed. Yeah, that's a real neat, let's see if I can find that on YouTube. So the seeds, the water potential of the seed is so low that when you make up the plaster Paris solution before it has a chance to evaporate and put the seeds in there and, make the mass, then it pulls water into the seeds start to expand and will crack, and the piece of plaster Paris will come apart. That's water potential. That's the energy. That seed starts to expand. It has nothing to do with whether or not the seed is living or dead. It's called imbibition, and that's when the seed pulls in all the water. Okay? Questions? So we have this pressure potential, we have this solute potential, and we have water potential. Can water potential ever be positive? No, I've told you several times it's 0 or lower. It can't be positive. So the pressure could be negative and negative. So these two components could be, we have a negative pressure, negative solute potential. We're always going to have a negative solute potential. We can have a positive pressure. So this turgid cell has a positive pressure potential. If it didn't have a positive pressure potential, the plant would be wilted. Okay? Now, in plants then, we're talking about mostly the vacuole, but we're not going to separate the vacuole from the cytoplasm. And we're talking about a positive pressure giving you turgor pressure, and the cell is turgid. And like I've told you several times, when the plasma membrane, the cell, the protoplast, presses against the cell wall. So if I go over here and put my hand, you guys know from physics that as I press against the cell wall, as I press against the wall's pressing against me, right? It's producing a pressure. So when that protoplast expands out, when you fill the balloon up with water and it fills up the wooden box, it produces a pressure. Now, if it doesn't, then the cell is considered to be flaccid. So, or in a leaf wilts, it can be considered to be the cells are flaccid. And then we have this term we call incipient plasmolysis. And this used to be used a lot more. This was something you could do with a microscope in cells. And incipient plasmolysis is when the protoplast is just filled, completely full, so the balloon is filled with water and it's touching all of the sides of the box, but it's not producing any pressure. Okay, it's not producing any pressure. It's not used much anymore because we have better ways of measuring water potential. So as we go to our cell here again, then let's see if this is working today. It seems to work about every other day. And it's not working today. So we have water coming in. Let me see if I can draw. So we have water coming in, which presses against which into the vacuole, which we then have water going out. But as this protoplast expands, and this is fully expanded, this was back in your notes then, it's going to press against the cell wall and it's going to produce a pressure. Okay? And then theoretically, like I've said over and over again, every time you have one water molecule move in, the pressure is going to push one molecule out. Let's talk about a couple other terms that we actually usually talk about in animal cells more than plant cells. At the left, you've got a thing called isotonic. Okay, the isotonic is when you've got pure water basically surrounding the cells, and they're trying. And in this case, these cells are just incredibly engorged with water, and they're pressing against the cell wall. In a hypotonic solution, the water potential is lower inside, and so the protoplast is no longer pressed against the cell wall, okay? And this color, by the way, is actually in the vacuole. Almost all colors that are not associated with photosynthesis are found in the vacuole, okay? Does that make sense? So it's isotonic or hypotonic. And I'm just going to pass that real quick. Some obvious things here. If you look at a wilted plant, then you know that the water potential of those cells in those plants are very negative, or more negative. I know more negative is not correct English. So you need to start thinking about in terms of the water potential being 0 or less than 0. Water potential is never going to be above 0. In the biological system anyway, or I mean, I'm sure you can force it. But anyway, okay? Questions? Okay, let's add another factor to this thing. Can you have a pressure potential without a membrane? And the answer is no. Okay, so if I have an open container here, and I put sugar in that open container, I've lowered the water potential because I have directly lowered the osmotic potential. Where's the pressure? There's no pressure, except for gravity, and we're not going to talk about gravity. So for a pressure potential to exist in a plant, it's got to be a membrane-bound something or other. Okay? It's got to be a membrane. So the pressure can be positive or negative. The water potential, solute potential are 0 or negative. Okay? So, questions about that? So you have where if I look at a tree, where's the highest water potential in that environment? Where, in order for the water to get out of the leaf in the sequoia, where is the highest water potential? Not in the roots. The soil. Water moves down a water potential gradient from the soil to the atmosphere. So it's complicated because it's just not water. It's solute potential, it's pressure potential, it's negative pressure potential in the xylem, pulling the water up. And remember, water moves up as a continuous column all the way till it gets to the vascular tissue in the leaves, and then it gets to the it gets into the leaf proper. Then it gets into the air spaces, and when it goes from a liquid in the cell to a gas in the air spaces, that's a tremendous decrease in water potential. And so then the lowest place you're going to find water potential is in the atmosphere, right outside the leaf. So if the water potential is not low outside the leaf, water can't move in the plant. Collectively move from the bottom all the way to the top. Okay? So let's look at how water moves in a system. In the xylem vessel, we're having the water potential is equal to 0. Okay? Now, that's water. It's got some stuff solubilized in it, and probably under normal conditions the pressure is actually a little bit on the negative side. So how do we move then water through a series of cells? Well, let's look at this. These are these values. The bottom line is that The water potential, the total water potential, is following this gradient. But we have different values of pressure here. Here, the pressure is 0.4. The osmotic potential is -5. So we have a water potential of -1. Here we have a solute potential of -4, a pressure potential of 0.2, and an overall water potential of -2. And in this third cell here, we have a solute potential that's much more negative. I know that's not correct. And a pressure potential of 0.2, and then a water potential of -3. If this had the same water potential, there would be no movement. So this is the direction of water movement. If we put a membrane in the system, then we measure the pressure and the solute components, and that explains how water moves. And there's all sorts of ways that we can measure this. Questions? Okay, let's see. Let's take the xylem out of here. Let's say I have 3 cells. And I put these 3 cells side by side so that their cell walls are connected, and I let them go. What's going to happen? Where's water going to move? So let's take the xylem out here. We have a theoretical perfect system where we have 3 cells that I've put in side to side so they're touching. What's going to happen? Which way is water going to move? Okay, so we're going to move to equilibrium. What's equilibrium? Do what? 0? Not 0. In those 3 cells, what's the equilibrium going to be? Is it? Let me look and see. It could be. What's it going to be? No. -0.2. Now, I've just said it's a joke because it's very common, extremely common when we go to seminars about water potential. No one ever says -2. They always say 0.2 because we all know it's minus, and it can drive you crazy. And you can go to a meeting, and there's several different ways of measuring water potential. So you'll have someone up there, showing their slides, and you'll have 20 people with their calculators trying to convert their terms into the terms that they use. So we know it's point. So when I say 0.2, it's -0.2. Pressure can be 0.2. So how's water going to move? Water's going to move back and forth till it's the same concentration in all of those cells. So if this is -0.1 and this is -0.3, they're going to move until all 3 cells have a water potential of -0.2. So water's just going to move back and forth. That's the only thing we're talking about is moving water. Okay? So in that theoretical system, -0.2 is going to be equilibrium, right?
Is it the average or?
The average, yeah, because if you think about, so water is going to initially move this way, right? Then the question is, well, how long does it move? Well, it's going to move until this is -0.2, because this is -0.2. So this is going to become -0. Get this thing 0.2. Okay, yeah, that's a 2. Okay, so then what's going to happen? Well, then a water molecule is going to move back this way. At the same time, this is going to move here, okay, till it's about 0.2. Then a water molecule is going to move actually then back this way, okay? Okay, let's finish up with really, and I don't know if I'll finish this today. We've talked about water moving passively. Now I'm telling you that there are these really, really important class of compounds. When they were first found, the guy that found them, they said, well, you're crazy. And these are a class of molecules called aquaporons. And they are found in, they're a major class of what they call intrinsic proteins. They're integral membrane proteins, and their job is to move water faster inside and outside of cells. So basically what they are is an integral membrane protein with a pore all the way through, and the amino acids in the integral membranes are arranged so that they pull water molecules through. They don't require energy, it's still passive, but they actually move water molecules through faster than water can diffuse through the membrane. And they will also move, in some cases, other neutral things like glycerol. Glycerol, if you know what glycerol is, it's basically a molecule that looks like water, 3 C's and no H's, so it sort of looks like water, so a neutral molecule. Very important. And so what they do is they can help to quickly regulate the water levels inside the different parts of the cell. Okay? So we have the cytosol, we have the protoplasm, and then we have the vacuole. And so these things can actually move water around, or help move water around faster. And they're called gated channels. Okay? What's a gate? Anybody know what a gate is? Nobody knows what a gate is? You know how many gates that you walk through? It's just like a gate, so they can open and close. So if the cell needs a lot of water inside really quick, these things will open up and sort of just like, fire hose water inside. Very important. Still trying to figure out exactly everything they do, especially in plants. We know a lot more about them in animals because they've got a lot more money. They're fairly complex. They span the membranes. They have several different domains. So what can you tell me about the polarity of this bit of protein? You had an exam on Friday and you can't tell me what the polarity of that would be? Huh? These proteins here are going to be nonpolar, right? They're basically soluble in the lipid part. So they're fairly complex proteins in animals, in plants. And they are also involved in osmoregulation, which we'll talk about next time, I think. And water can move both ways. Here is in this case. And in here we have this channel protein. So water would move actually faster this way. And then finally, here's, I want to put this up. This is from Wikipedia. Who also knows it's not in English. But it's a diagram of how the structure of the actual opening helps to move water. And that's because the proteins, in this case, here's this protein 180, here's protein 192, so they're pretty closely linked proteins, but they're made in such a way, and then on this other side we have a protein which nitrogen, 76? And you notice they're arranged in this three-dimensional shape that produces these positive charges, and that sort of helps pop water through here. So it grabs a water molecule, and as it does, it changes shape, and that pushes a water molecule down. So it's not covalently grabbing it, but it's just because of those amino acids is sort of pushing the water molecule through. Okay? Very important. They've not been around that long, and so we know, they probably could be more important in plants than they actually are in animals. Okay, questions? Okay, we're finishing up. See you next time. Oh, goodness, I forgot to put the hell,